Hi, y'all. So last class we talked about DNA replication. We did some practice with it with the marshmallow and the Twizzlers. And then we also talked about interphase, which is the majority of the cell cycle. It's like 90% of the cell cycle. I know you can't do the do now in your journal, so I want you to at least think about it. And so the question says, we already talked about the interphase parts of the cell cycle last class. Now, why is the mitosis part of the cell cycle so important? Can mitosis happen without interphase? These are some good questions to think about, so take some time. You can pause the video if you need to. Now, I want to be clear, mitosis is the part that we're going to talk about today, and it's the stuff that happens after interphase. So just knowing that it happens after interphase, you can already take a good guess that, yeah, you need to have interphase happen first because the cell grows, it duplicates organelles, the stuff inside the cells, and then you're also going to be duplicating the DNA or replicating the DNA. You need a set of DNA for every single cell. So when you're splitting the cell into two new cells or two kind of half new cells, they both need the full set of DNA in them. So that all has to happen before the cell even splits, which is what mitosis leads into. And that's what makes interphase so important. The objective for today is going to be students will be able to explain the importance of mitosis in the growth of organisms by describing prophase, metaphase, anaphase, telophase, and cytokinesis within the cell cycle. So those first four PMAT phases, those prophase, metaphase, anaphase, telophase are all part of mitosis. And then it's followed with cytokinesis, where the cytoplasm for cytokinesis happens and splits into the new actual cells. So this is some important stuff, so please pay attention. You're going to be filling in your notes packet from the rest of last class along with this, and then you're going to have two things that you're going to work on at the very end that you should already have grabbed before you left last class. If you didn't, please make sure you come grab it before you watch the rest of this video. So finishing the notes, you want to draw a chromosome. It's this weird X-shaped bubble letter, kind of like a X figure, and you're going to label that each side is a chromatid. They match each other, so they're considered sister chromatids. And in the middle, in the center, there's a centromere. It's named exactly what it's called, right? It's in the center of the chromosome, so it's a centromere. Haha, very creative. So chromatin is this like noodly spaghetti stuff that your DNA normally is in the phase of inside of your nucleus in your cells, and it's really messy. So it's kind of hard to pull chromatin apart from each other to make sure that you get an even split 50-50 into the new cells. So we coil those chromatin noodles into chromatids, which are each half of a chromosome. Each half is identical to the other half, and these will be splitting off to become, instead of two X's kind of touching in the middle like this, they'll become their own little lines or their own little chromatids by themselves. The centromere is merely the part in the middle where they're connected, or in the center. If you recall, you should know how many chromosomes humans have. A sperm and an egg each have 23. So when you add them back up together, for a total human genome, you should have 46 chromosomes. Hopefully you remembered that. Mitosis is the short process that happens for about 10% of the lifespan of a cell. It happens after that 90% of interphase, because interphase is when the cell is just like existing and doing its job. And then on the by side, it's replicating DNA. It's growing organelles. It's growing in size. But mitosis happens in a snap of a moment compared to the whole rest of interphase. It's when the actual parent cell is splitting into the two daughter cells. And in order to do that, it needs to first split the nucleus into two nuclei. Prophase is the first part of mitosis. It's the P in PMAT. In prophase, the chromosomes will form, the centrioles will separate, the mitotic spindle begins to form from the centrioles, which are these long fibrous strands, which will then eventually connect to your chromosomes and pull them apart. Now, because there is a nucleus on the outside of these chromosomes protecting them, it's kind of getting in the way. So we're going to want that nuclear envelope to break down or start to dissolve. So during this time, the DNA is not really protected. So mitosis has to happen really fast, because if you leave your DNA uncovered, it actually is really easy to damage it and cause mutations. So again, as I'm describing this, you can either draw this image or you can pause the video to draw this image in the space where you're provided for prophase. So one more time, the chromosomes are forming from chromatin. It's going from that noodly form into those bubble letter X's. So it's really easy. It's like grabbing like dolls and pulling them apart instead of grabbing spaghetti strands and trying to evenly pull them apart. The centrioles are these things that actually only animals have, and they will separate after they form and go to the opposite end slowly and form their spindle fibers, which will eventually connect to the chromosomes once the nuclear envelope has fully broken down. That's it for prophase. Moving on to metaphase. Metaphase is the M in PMAT. And metaphase, the M is really also easy to remember because M stands for middle. As you can see, all these chromosomes are lined up in the middle of the cell. So M for metaphase, M for middle. I would highly suggest writing down the word middle next to the word metaphase on your page. In metaphase, as you draw this out for yourself, the chromosomes will line up across the middle or the center of the cell. You can see that it's a single file line. Each chromosome is then grabbed by a spindle fiber at the centromere. So the centers of those little chromosomes that we were talking about before are the access point for the spindle fibers to attach, kind of like a spider web attaching onto a fly or a bug. And then eventually they're going to pull them apart. But that's not just now. In metaphase, they're just lined up across the middle. And the spindle fibers kind of help them make sure they're lined properly. You don't want them overlapping. You don't want them doubling up. They want to be in a single file line in the middle. So again, M for metaphase and M for middle. And that's the M in PMAT. On to anaphase. Anaphase is the A in PMAT. And the A also stands for away or apart. As you can see in this picture, the chromosomes used to be an X shape, but now they're just a capital I shape because they've been pulled apart or away from each other by the spindle fibers. So the chromatids are now separated. We no longer have chromosomes, just chromatids separated, and they're being pulled apart in anaphase. So anaphase is also a pretty straightforward one to remember just because A just means away or apart, and you can literally see in the picture that chromosomes are being pulled apart as chromatids to the opposite ends. Some pictures you'll see they're really just barely starting to be pulled apart, and some pictures they'll be all the way on the opposite ends already because they've been pulled really far apart. So both those phases or those picture versions would be of anaphase though, because you see the chromosomes being pulled apart into the chromatids. Last step in PMAT of mitosis is T for telophase. And what I always say is, even though I didn't write it here, telophase, T, there's two nuclei. As you can see in the picture, there's a bubble here and a bubble here. And the chromosomes are no longer those X shapes. They're back into their noodly forms of chromatin as they kind of become disorganized again and become their regular form that they're about to go back into interphase. And so the chromosomes are now fully separated from each other, and the nucleus is forming in each of those new cells that are going to form. Okay. So in telophase, you have two nuclei. That's the easiest way to remember. And that's the end of mitosis, or PMAT, PMAT. Make sure this is drawn. I know the shape on your paper is a little bit different from this. So you can draw the general idea of this into those two separate little sides of the long circle, but you can draw the little divot inwards as well if you would like to, so that you can show that, you know, it's over and the shape is different. So it's kind of obvious that it's over. The last thing that happens after PMAT is cytokinesis. And like I explained earlier, cytokinesis has the word cyto in it, which literally refers to the cytoplasm. It's the same word, okay? Cytokinesis is the movement of the cytoplasm. Kinesis means movement. Cyto means cytoplasm. So the cytoplasm is going to kind of squeeze down in the middle. And eventually it squeezes so far that the ends actually touch from the center and it fully separates as the membrane meets itself in the middle. So the cytoplasm is pinching in half, and then you get two new cells. Obviously, you want one nucleus in one and one nucleus in the other, because if you did this whole process of PMAT and then you put the two nuclei in one, then you would have a deformed cell, and then you would have a cell without any nuclei, which means it would die, okay? So you want the cells to each have one of the nuclei that you separated in PMAT, and you're going to actually want the cells to separate into two separate cells, because if they stay conjoined, then they're not really serving their purpose of replacing or helping you grow, okay? So cytokinesis is the physical separation of the cell, whereas PMAT was just the separation of the DNA, the nucleus, okay? Cytokinesis in plants versus animals is different. In animals, like I said, you get that pinching where there's a ring that forms and it pinches in until it meets in the center. And then in that center, the two cells can then separate because they've touched in the center. So now they're fine. But plants, they have a cell wall on their outside. And because the cell wall is stiff and hard and supportive, you can't just pinch the cells inwards because there's resistance. So instead of doing that, they grow something called a cell plate in the middle of the cell. And the cell plate kind of starts out spotty in the middle. And as it expands outwards, eventually it meets all the way to the ends of where the cell wall originally was. And it forms a brand new cell wall there in between where the two nuclei are. And in doing so, once it's fully formed, it separates those two halves and they become separate cells in a plant. Those cells will go on to grow and become the normal size of a cell again in a plant. But for now, they're a little bit smaller. There are some questions that I do want you to refer and fill out in your packet. That way I can evaluate whether you are paying attention to this video. The first question is, interface is broken down into 3 phases. What are those 3 phases? This is actually a throwback from our last class together, so please make sure you either look at those notes or you just write them down if you remember them. The second question asks you, during which phase does DNA replication occur? This is also an interface question. What might happen if the checkpoints in the cell cycle did not exist? There's checkpoints across the whole cell cycle, so you should know in general what happens if they don't exist. And then lastly, why does the cell cycle exist? Interface is broken down into 3 phases. Those phases are G1, S, G2, growth 1, DNA synthesis, and growth 2. During which phase does DNA replication occur? That's going to be an S phase because DNA synthesis literally means DNA is being made or DNA is being replicated. What might happen if the checkpoints did not exist? Well, there would be no way to check if you just did the last step or the last phase correctly. So you'd have a bunch of mistakes in your cells, a bunch of mutations, likely cancer. Cells would divide when they're not supposed to. Cells maybe wouldn't even divide when they're supposed to. So a lot of issues could happen if there were no checkpoints to make sure that we're all good before we move on to the next phase. And then why does a cell cycle exist? Cells have to grow, cells have to reproduce, cells have to replace other cells that are dying, right? Cells have a lifespan. It's much shorter than our entire lifespan. So there's cells that are dying in your body right now and cells that are replacing them. So there are plenty of reasons why cell cycles need to exist for us to get more cells. What you're going to do now is we are finished with the notes. Again, I will check those questions, so make sure that you go ahead and have those filled out, whether or not you did them on your own and you corrected them, or you didn't know them really and you got to kind of get the answers from me just now. The mitosis video, I'm going to have you watch and fill the handout as you go through. Try to get it done by watching it 2 times through and no more. If you need to, you can pause as you go to and rewind as much as you need to during those 1 or 2 times that you're watching them, so that you're not wasting too much time on it and you're making sure that you get all the information done. Okay. The handout is the Amoeba Sisters handout. And I'm going to play the video on here as well if you need it. But it's linked inside of your Canvas, so you can use that one as well. Let me share the sound from here.
Have you ever looked down at your leg or your arm to find some cut and you have no idea where it came from or how you got it? So you put a bandage on it and a few days later it's gone. It's all healed and you don't even think anything of it, of this amazing process that causes this to happen. Or let's say you're looking at your nails and you notice they're a lot longer since the last time you cut them. Or let's say you're looking in the mirror and you notice that you are a lot bigger than you were when you were 5 years old. What do all of these things have in common? Well, one major thing they have in common is mitosis. Mitosis is a type of cell division done by most of your body cells, and it's really important for cells to divide. If they didn't divide, you wouldn't grow. I mean, how do you grow if you can't make more cells, right? So one reason why you're bigger than you were when you were 5 is mitosis. Mitosis also is great for repair of damage. If you have some kind of accident, like when we were talking about that cut on your arm or leg, well, you want to make sure it gets repaired well. So you have to make more cells to do that. Mitosis is great for that. Now, it's really important to understand what it is not. Mitosis is not a process that makes sperm or egg cells, because that's something different called meiosis, which sounds like mitosis, unfortunately, but it's a different process. Mitosis is done to produce body cells. Mitosis makes identical cells. That is the goal, identical cells. So if you're trying to make more skin cells to replace worn out or damaged skin cells, you don't want to start suddenly making stomach cells there. That would be ridiculous. You want to make sure you have identical cells replacing what was lost. So mitosis makes identical cells. It's a really important thing. Now, it's also important to understand that your cells are not dividing all the time. If all they did was divide, it would just be rapid, crazy growth. In fact, this is kind of what cancer is. Cancer is uncontrolled cell growth. We have a clip on the cell cycle and what the cell is usually doing most of its daily life, which is actually a phase called interphase, where it's growing and replicating its DNA and carrying out its daily cell functions. That's where cells spend most of their time in respect to the whole cell cycle. Mitosis is a very short amount of time in respect to the whole cell cycle. But mitosis is a critical process because this is where it is going to divide and make more cells. Before we get into the steps of division, it's really important to understand that your cells have something inside them, an organelle called the nucleus. And the nucleus holds your DNA. DNA is really important because it's your genetic information. And if you're going to make more cells, you need to have the same DNA in those new cells as you did in your original cells. You want it to be identical, no mistakes. Very important. The problem is you've got a lot of DNA, and we've got to get that DNA into the new cells using mitosis. So there has to be a better way to organize that DNA. Well, what actually happens is that DNA can be organized into these condensed units called chromosomes. Chromosomes are made of DNA and protein. You've probably heard before that humans have 46 chromosomes. That means 46 chromosomes are found in most human body cell nuclei. What are nuclei? Well, it's the plural of nucleus. You don't say nucleuses, you say nuclei. Well, in the nuclei, there are 46 chromosomes. Organizing DNA into condensed chromosomes makes it a lot easier to move over when you're making new cells. So if you have 46 chromosomes in a human body cell, you have to duplicate those chromosomes in interphase before mitosis starts. That basically means you're duplicating your DNA since chromosomes are made of DNA and protein. You have to do this before mitosis starts because if you're going to make an identical cell that has 46 chromosomes just like the original, well, it makes sense you have to duplicate the genetic material before splitting. So if you look at our cell cycle video clip, we talk about interphase. That's a stage where most of the time cells are spending their time. They're actually duplicating their DNA during that time. So ready for the tricky part? Well, because we tend to count chromosomes by the number of centromeres present, when the 46 chromosomes duplicate, we still say there are 46 chromosomes, as the sister chromatids are still attached. And we're counting by centromeres. So 46 chromosomes here, they replicate in interphase, and you still have 46 chromosomes in this picture, but you went from 46 to 92 chromatids. We have a video explaining that in more depth and how that factors in for mitosis. Okay, so now we can get right into mitosis. I like to tell students to remember PMAT. It's a little acronym that helps you remember. The P is for prophase, the M is for metaphase, the A is for anaphase, and the T is for telophase. So remember PMAT, the stages in order. The very first step is prophase. Prophase because it's the beginning step, the nucleus is still there, and it's going to go away later on, but this is a stage where it's actually still there. The chromosomes are visible. In fact, we say they're condensing, which means they are thickening and visible. The next stage is metaphase. M for metaphase, but I also like to remember M for middle because in this stage, the chromosomes line up in the middle of the cell. The nucleus has been disassembled. It's no longer there, so we've got the chromosomes in the middle waiting there. Next, the A is for anaphase. In anaphase, I like to think as the A for away. The chromosomes move away. They are moving to opposite sides of the cell, so they are moving towards the poles of the cell. Now, one thing to point out, these chromosomes, they're not moving by themselves. They actually have something called spindles. These spindles are fibers that help move the chromosomes to the ends. Kind of helps them move along. The last stage of mitosis, think T for telophase. In telophase, the chromosomes are actually at the complete opposite ends, and new nuclei are forming on each side to make these two new cells. The nuclei are starting to surround the chromosomes on both sides. I like to think of the T for 2 because you can really see in this step that the end goal is going to be 2 cells, and in the human body, they're each going to have 46 chromosomes. And again, remember, they are identical. Now, cytokinesis is responsible for the final separation into 2 cells by splitting the cytoplasm, which completes after the PMAT mitosis stages. So why did all this matter? Without understanding cell division, we wouldn't understand how growth and repair happens, because they both require more cells to be made. Understanding mitosis is also very important for cancer research. Cancer itself is uncontrolled cell growth. So, in other words, uncontrolled mitosis. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
All right, so for that video, you can watch it on here and repeat it as much as you need to for the sections to get all those answers in your worksheet done, or you can watch it from the actual link inside of Canvas. It's up to you. It doesn't really matter which way. After you watch the video, you're going to do the coloring sheet, which is on the back. If you don't have access to colors, you can always drop by my room to pick them up at any time. And make sure you return them. But if you want to use your own coloring pencils, that's totally okay. If you don't have the exact same colors, just scratch out the name of the color that's on the paper and replace it with one that you do have. I have no issues with that. I just need to make sure that you get all of that front side done. The back doesn't require any coloring pencils, so you only need the coloring for the circle diagram on the front. Okay, once you're done with the coloring and the video worksheet, you're going to turn that packet into me. You can turn it in later today if I don't have you in the morning because of testing, or you can turn it in the next time I see you, which will be in another school day. So once you're done with all that, make sure you turn it into me again so that you can get your graded stuff, and I'll see you on Friday.